Welcome to this lecture on the fundamentals of genetics. Today, we are going to explore a fascinating and crucial topic known as the linkage map. If you have ever wondered how scientists figure out exactly where specific genes are located on a chromosome, or how they track down the genetic causes of inherited diseases, you are in the right place. We will build our understanding from the ground up, starting with simple concepts and moving all the way to calculating actual genetic distances. Let us dive in and decode the map of life together. To understand how linkage maps work, let us start with a simple analogy. Imagine you are looking at a map of a very long highway with several towns dotted along it. If two towns are located very close together, it is highly unlikely that a random roadblock or an accident will happen exactly between them. However, if two towns are very far apart, the chances of a roadblock occurring somewhere between them are much higher. In genetics, a chromosome is exactly like that long highway, and the genes are the towns located along it. This simple idea of distance and probability is the entire foundation of genetic mapping. Now that we have our highway analogy, let us look at what actually happens in the cell. When cells divide to make sperm or eggs, chromosomes often swap pieces with each other. This process is called crossing over. These crossover events act exactly like those random roadblocks we just talked about. If two genes are far apart, a crossover is very likely to happen between them, separating them into different offspring. By carefully measuring how often two genes get separated by these swaps, we can figure out exactly how far apart they are on the chromosome. This is how we begin to draw our map. Building on that idea of crossing over, let us formally define what a linkage map is. A linkage map, which is also known as a genetic map, is a linear diagram showing the relative positions of genes on a specific chromosome. It is important to distinguish this from a physical map. A physical map measures the absolute distance between genes in actual base pairs of DNA, much like measuring a road in miles. A linkage map, however, measures distance based entirely on recombination frequencies, how often those genetic roadblocks occur. This gives us a functional map of how traits are inherited together. Now that we know a linkage map uses recombination frequencies, let us define what that means. Recombination frequency is simply the percentage of offspring that inherit a new, mixed combination of alleles due to crossing over during meiosis. The fundamental rule here is that the probability of a crossover event occurring between two genes is directly proportional to the physical distance separating them. We measure this genetic distance in units called centimorgans, abbreviated as cm, or simply map units. By definition, 1 centimorgan is exactly equal to a 1% recombination frequency between two genes. Before we move on to the math, we must understand why this matters in the real world. Understanding how to create and read a linkage map is absolutely crucial in medicine and agriculture. Before modern DNA sequencing was invented, linkage maps were the only way scientists could figure out the architecture of a genome. Today, they are still heavily used to track down the genetic causes of inherited diseases in human families. If a known marker gene is always inherited along with a mysterious disease, doctors know the disease-causing gene must be located very close to that marker. Furthermore, agricultural scientists use these maps to breed better crops, saving years of trial and error. Now that we understand what a linkage map is and why it is important, let us look at how we actually calculate these distances. To build a linkage map, we set up a specific mating experiment, usually a test cross, and count the offspring. The formula for recombination frequency is the ratio of recombinant offspring to the total number of offspring, multiplied by 100. The variable RF stands for recombination frequency. The numerator, NR, is the number of offspring that inherited a new combination of traits due to crossing over. The denominator, NT, is the absolute total number of offspring produced, including both parental and recombinant types. If the number of recombinant offspring increases, the RF value increases, indicating the genes are further apart. With our formula ready, let us see this with a concrete example involving fruit flies. A scientist crosses a heterozygous fly with a homozygous recessive fly. They count 400 offspring with parental phenotypes and 100 offspring with recombinant phenotypes. Let us solve for the genetic distance. First, we find the total number of offspring by adding 400 and 100, giving us 500 total flies. Next, we substitute our values into the formula. We place the 100 recombinant flies in the numerator and the 500 total flies in the denominator. Dividing gives us 0.20. Finally, multiplying by 100 gives us 20%. Because 1% equals 1 map unit, the genetic distance is exactly 20 centimorgans. Now that we can calculate individual distances, how do we map multiple genes? We use the additive property of distances. Conceptually, if 3 genes lie in a straight line on a chromosome, the distance between the 2 outside genes must equal the sum of the distances between the adjacent inner genes. Here is the formula. DAC equals DAB plus DBC. The variable DAC represents the largest distance, which separates the 2 outside genes. The variables DAB and DBC represent the smaller distances between the adjacent genes inside that segment. If the physical distance between the outer genes increases, the sum of the inner segments must increase by the exact same amount. Let us apply this formula to a real scenario. We are given 3 genetic distances for genes A, B, and C, and we need to find their order. The distance between A and B is 12 centimorgans. The distance between B and C is 5 centimorgans. The distance between A and C is 17 centimorgans. To solve this, we simply look for the largest value. The largest distance is 17 centimorgans, which belongs to genes A and C. Therefore, A and C must be the outside genes on our map. This tells us the physical boundaries of our gene sequence. Now, we must mathematically prove that this sequence is correct. Since A and C are on the ends, gene B must be situated between them. We verify this by plugging our numbers into the additive formula. We add the segment DAB, which is 12, to the segment DBC, which is 5. 12 plus 5 equals exactly 17. This sum perfectly matches our largest measured distance, DAC. By showing that the smaller segments add up perfectly to the largest segment, we mathematically confirm that these 3 genes lie in a straight line. We have successfully proven that the correct linear order is A, then B, then C. Now that we have proven the order mathematically, let us look at the final diagram of our 3-gene linkage map. You are seeing a thick horizontal line that represents the chromosome itself. Along this line, there are 3 distinct points marked A, B, and C. Notice how gene B is placed between A and C. Above the line, you can see the individual distances we measured: 12 centimorgans separating A and B, and 5 centimorgans separating B and C. Below the line, a larger bracket shows the total combined distance of 17 centimorgans from A all the way to C. This visual perfectly captures the additive nature of genetic distances we just calculated. We have covered a lot of ground today, so let us review the key takeaways. First, a linkage map is a powerful genetic tool that translates the frequency of crossing over into physical distances on a chromosome. Second, by calculating recombination frequencies from breeding experiments, we can determine exactly how far apart genes are, remembering that 1% recombination equals 1 centimorgan. Third, by using the additive property of these distances, we can determine the sequential linear order of multiple genes. You have now learned the foundational step in isolating genes responsible for specific traits or diseases, bridging the gap between abstract inheritance and actual physical DNA. Great job following along.